welcome to the class Advanced Reaction Engineering. So, today we will discuss about bioreactors. So, that is basically cell growth and rate laws that we will discuss. So, in our previous class or at the beginnings we have discussed about the bioreactors. So, what is called bioreactors and how does it look like. So, here you can see a schematic diagram of a bioreactor that is given here. So, this is one kind of batch reactors you can say. So, here in this type of reactors air will be supplied here. Here's this yellow portion, that is the filling port, that is the acid, base or any nutrient that would be supplied for the reactions, or the cell growth, the cell will be supplied in that particular inside of the reactors. So, through that inlet. Inside of the reactor there is a one agitator is also there. This is called agitator to mixing the reacting mixtures when reactions will take place at a particular temperatures. And outside you can see there is a cooling water supply is there, cooling water. So, to maintain an isothermal temperature for that bioreactor operations. Also a spurger is there. So, you can see here would be supply that would come, and from the bottoms in the form of bubbles that would be dispersed to the liquid. So, for the reactions, once the reactions will be over, then effluent we can come out from the bottoms or any other ports. That is the schematic basic diagram. So, all these reactors, that is bioreactor means it is reactor that sustains and support life for cells and tissue culture. All cellular reactions necessary to maintain life are mediated by enzymes, as they catalyze various aspects of cell metabolism, such as the transformations of chemical energy and the construction, breakdowns and digestion of cellular components. The enzymatic reactions are involved in the growth of microorganisms. Therefore, we need to study the microbial growth and bioreactors. The Monod equations describe the growth law for the number of a bacteria that is present or that is means grow inside of the reactor during bioreactions, and it is also similar to the Michaelis maintenance equations. The growth of the biotechnology, if we look into that, so bioreactors are not truly homogeneous because of presence of living cells. The use of living cells to produce the marketable chemical products and is increasingly day by day, and it is very important to produce the particular product that is from a bioreactions or from the bioreactors. So, chemical, agricultural products, food products are produced by biosynthesis. Many products are there that is synthesized from bioreactors. The microorganisms and mammalian cells are being used to produce a variety of products such as insulin, antibiotics and polymers. The advantage of bioconversion is mild conditions with a high yield. When we operate a bioreactor, it requires very low temperatures. So, easily we can maintain. So, that is the advantage of use of a bioreactor. And for example, we can say if we want to get glucose to gluconic acids with Aspergillus niger, that is the bacteria we can use, here the conversion would be 100%. So, that is the advantage of bioreactor using mild condition 100% yields we can get. Bacteria can also be modified and turned into living chemical factories. In biosynthesis, basically the cell is transferred to as the cell is referred to as the biomass. In the biosynthesis or in the inside of the bioreactors, the culture we are doing, that is the basically biosynthesis. In that process, the cell whatever we are using, that is as a biomass, and that consume nutrients to grow and produce more cells that are important. Also more important products are also formed during that process or the biosynthesis process. So, internally a cells use as the the nutrients that is given for that particular reactions to produce energy, at the same time to produce more cells. The transformations of the nutrients to the energy or byproducts is accomplished by a cell use of number of different enzymes in a series of reactions to produce metabolic product. So, here you can see I am showing here diagram. So, here you can see the product, how the product may forms or the schematic of a cell. Here you can see this is a schematic of the cell. So, here this is the cell wall, this is the cell wall, this is the cell membrane, and so here we can supply the nutrients will that is inside of that cell, and this is we can say ribosomes or cellular cell nuclear region is this. And the after let us say once a cellular reaction is there, so when the cells consumes some substrate, then it forms some products maybe. So, that is maybe CO2, waters or any other product that may forms. Okay. At the same time the cells will grow or in the multiple numbers. So, sometimes the product that is forms that may be inside of the cells or may be outside of the cells. If that product is present inside of the cells, then we have to rupture the cells, and then we can separate out that particular product. If we have interest or that have value, then we can separate out that product, or maybe product maybe after reactions, maybe the product we can get in the reaction mixture, that also we can separate out. Okay. So, here when the cell reactions is taking place, means we are supplying the nutrients. There are we can classify the cell reactions as class 1. So, that is the nutrient degradations, means fueling of the reactions, means when a particular nutrient that we are supplying for the cell culture. So, then that is the nutrient degradation, nutrients degrade, and the second class is the synthesis of the small molecules or amino acid kind of things that will be formed, and then third is the synthesis of the large molecules, means polymerization reactions, DNA, RNA formations. So, here you can see the reactions that will take place or reactions occurring inside of the cells. So, nutrients that we are supplying, that is enter into the cells, that nutrients will consumed by the cells, and then some reactions will be there, some small molecules, that is the amino acid formations would be there, and further that would be converted into proteins or ribonucleotides, RNA or DNA. Further polymerization reactions would be there, and larger molecules formations is there. So many reactions are there, means step by step from nutrients that is consumed by the cells, and so many reactions are taking place. So, here you can see the cell growth and cell divisions, means the cell growth and cell divisions. Typically the mammalian cell that is given below, means there are 4 stages are there for the cell divisions, that is G1, S, G2 and M. Okay. So, these are the 4 different stages we can say, or 4 different phases cell divisions are take place. So, here with the times, let us say at the beginning to at any times t. So, means if we start with a new cell, so then G1 phase is there, then S phase, G2 phase, M phase, and then new cell formation is there. This is the from 0 time to at any times the cell, how the cell divisions are there. So, new cells when it is there, so cell increase in size. So, in the G1 phase, first phase is the G1 phase. At this phase cell increases its size, RNA and protein synthesis will take place at this stage, but no DNA formations or DNA synthesis will take place. In S phase their DNA doubles, here you can see the DNA that is doubles, RNA and protein synthesis will occurs at this stage, and G2 phase here also RNA and protein synthesis will take place, but no DNA synthesis will take place. So, after that in M phase formation is there, so here the mitosis or nuclear region divisions is there, you can see. So, after that in the second M phase here cytokinesis, cell divisions will take place and two new cells will forms at this way. This is the way in the bioreactors when we are supplying taking any new cells and we are supplying nutrients, then cell divisions is there and new cell growth will take place. So, basically if we think about it that how multiplication is there or growth cell in the an aerobic organism follows these steps: cells, carbon source, nitrogen source, oxygen source and phosphate source. These are the things or the nutrients with the cells we have to supply inside of a bioreactor, and after that we if we maintain particular temperatures and pH inside of the reactors, then new cell formations would be there. At the same times few products formations would be there, byproduct formations is there. So, byproducts may be CO2, water, these are the byproducts, and new product formations would be there. At that products may be useful for a particular application. So, these products also is very important. So, with the more cell culture this product formations is also more important. So, next is the if we abbreviate the cell reactions inside of a bioreactors, here you can see we have taken the substrate inside of the reactors, at the same times we have taken few new cells. So, then after if we maintain pH temperature inside of the reactors, so more cell growth or more cell formations would be there, at the same times product formations would be there. So, actually the cells converted into more cells by consuming substrate, at the same times product formations is there. So, here one example is given, you can see glucose was the substrate and new cells that Saccharomyces cerevisiae that was taken as a cells, we forms more cells and ethanol, that is the product formation is there. So, in this particular reactions ethanol that was product formation was from glucose to ethanol formations. We have used the cells, particular these cells we have used to conversions of glucose into ethanols. So, here not only ethanols, apart from ethanol there are some other reactions also there, bioreactions, there are some valuable products formations as there, those are very important. Okay. So, here the substrate already I have discussed about, these are the nutrients that is required for the cell reactions, and that product that would form CO2, waters, proteins or other species that may be formed depending upon that particular reaction that we are conducting inside of a bioreactors. So, here the rate of the means when we are talking about the bioreactors, bioreactions, then the rate of the reactions that is very important, that strongly or proportional that is directly proportional to the cell concentrations inside of the cells that is present. So, rate of the reactions that is directly proportional to the cell concentrations. So, here when we are talking about the cell growth, so you already have studied. So, just I am discussing the basic things. So, here let us say we have taken in a container. So, there's or a batch reactor you can say, there at t equal to 0 time we are putting small number of cells that we are putting or that we are mixing here. So, after that if we wait for t equal to t a time, so then the cell concentrations at different stage you can see the cell growth is there, number of cells gradually increase. Okay. And after that a certain period will come, so then we can see the number of cell concentration decrease. Okay. So, in the when the cell concentration gradually increase, that is called lag phase or phase 1. In the second phase, that is the growth phase, and third one is the stationary phase. Okay. So, means this is the cell reactions in presence of substrate that we are supplying for the cell reactions in presence of cells, that is converted new cells, and at the same time product formation is there. So, in the right hand side you can see x axis we have taken times and y axis cell concentrations, that is in log scales if we plot. So, you can see here from here to here, that is phase 1, this is phase 2, this is phase 3 and this is phase 4. So, phase 1 that is the lag phase. So, when we supplied the substrate and cell in a new reactor or new environments, then it takes time to adjust the cells to adjust with the new environments, and at this stage the growth is very slow or negligibly very small you can say, and in the second stage, that is the growth phase you can see. So, here exponentially it is increased, the cell number of cells in presence of nutrients, the number of cells gradually increase, and after that in third phase. So, then the cells grow, means number of cell formations, that is there is no increase, means the number of cells may be there that present there. So, whatever the number of cells that have been formed during the reactions in the this growth phase, so that remain there. So, after that, and this phase is called actually stationary phase, this phase is called stationary phase. After that stage 4 means number of cells gradually decrease, that is you can say a death phase, or means cells death is there. So, number of cells counts that would gradually decrease in that particular point. Okay. So, here you can see, so at phase 1, that is the new environments at this particular point, the cells take times to adjust with the new environments for the growth, okay, and cell is ready for growth. So, this is called exponential growth phase for the cells that are able to use the nutrient that was supplied in the bioreactor. So, that would be used and number of cells gradually increase, and in here in the third phase you can see this stage is called means stationary phase, or stationary phase you can say this particular phase. So, here there is no number of cells formations stopped. So, that may be many reasons. So, one of that may be lack of one or more nutrients, that is we have supplied, that may be a lack was there, means unavailability, that is why cell growth, further cell growth has stopped. So, or maybe important fermentation products or antibiotics that are forms that inhibit the cell growth. So, maybe number one is reason is that nutrients unavailable, and number 2 maybe the product that is formed that inhibit the cell growth. So, therefore, here cell growth is stopped, no new cell formation is there, but the what are the cells are present there that sustain, and for that nutrients we have to supply, or that would be present inside of the reactor, they will consume. So, after that this is the death phase you can say, means number of cells that was present there. So, with increasing further time the number of product formation gradually decrease, that may be the toxic product that I said. So, that was formed here in this reactions you can say ethanol product formations is there, that is product that forms. This ethanol is the product in this glucose 2 in this particular cell reactions, more cell formation is there, but ethanol as a product formation is there, that ethanols may act as a toxic materials to degrade or for death of that new cells. Okay. So, therefore, cell number of cell concentration gradually decrease at this stage. So, now we know different phases of cell means from cells and nutrient to the number of cell formations and then cell growth, that would be these are the different stages with y axis, that is the concentration of the cells, and x axis the time that we have discussed. So, now we have to develop some rate equation, since we are using bioreactors. In the bioreactors the cells we are used and substrate that we are supplying for the cell reactions, and after the reactions cell formations would be there. At the same times the product formations would be is there, that product may be useful for various applications. So, therefore, once we study these cell reactions, then we should know the rate law for that bioreaction systems. Okay. So, here's monotype of, here you can see at the beginning, let us say NC amount or gram of cell that was taken in a reactors, that may be batch reactors, it has a particular volume. So, that much number of cells that was taken at t equal to 0, or cell concentration Cc, that you can say gram cell per dmq, dmq is the volume. So, per unit volumes the gram of dry cell that is present inside of the reactor, and so once time is passed in presence of nutrients, this type of reactions will take place inside of the bioreactor, then number of cells formations will increase. So, then concentrations we can express a gram of cells per dmq, per unit volume of the reactors. So, it is increase. Okay. So, this is the cell reactions basically happen. So, for that we have to find or develop some rate equations for that cell culture or cell bioreactors. So, here monotype of equations is used. So, I said here the cell growth that usually observes in the stage 2, this is the exponential stage growth, and here at this third stage there is no new cell formations is there, but for this bioreactor stage 2 and stage 3 both are very important. Okay. So, stage 2 here number of cell formation is there, at the same times maybe product formations is there at this stage, and most of the reactions bioreactions have been observed that third stage. So, when there is no new cell formations is there, at that stage new product formations or byproduct formations is there at this stage. Okay. So, therefore, these two stages are very important. So, we will discuss basically at these two stages, okay, growth phase and these stationary phases. So, let us say here the monod equations you can see for the exponential growth of cells. So, that is expressed in terms of Rg mu into Cc. So, Rg is the rate of cell growth rate, Rg is the cell growth rate and mu is the specific growth rate constant you can say, or Cc is the cell concentrations. Cell concentrations expressed in terms of gram cell per dm³ that reactor volume, and specific cell growth rate it has units second inverse or time inverse, and Rg is the growth rate that you can see here concentrations gram per dm³ per unit times. So, Rg is the monod equation that is for exponential cell growth. Okay. Rg equals to mu into Cc. So, this way you can express. So, now the specific cell growth rate that is the mu. So, that we can write or we can express this way, mu equals to mu max into Cs by Ks into Cs. So, here mu max that is the maximum specific cell growth rate, and Cs is the substrate concentrations or nutrient that we have supplied for the this reaction. In the left hand side you can see we have substrate and cell, two components are there. So, substrate, what would happen when bioreactions will take place, substrate concentration gradually decrease and cell concentration gradually increase, at the same time product concentration also gradually increase, okay, and more number of cell formation is there. So, here mu equals to mu max Cs, that is the substrate concentrations, into Ks, that is the monod constants, Ks is called monod constant and Ch is the substrate constant. So, mu we can substitute here. Okay. So, then monod equations for the exponential growth of the stage 2. So, here in the stage 2 that growth of the cells, that is the exponential growth. So, we can express this way. Okay. So, Rg equals to dCc by dt, concentrations of cells, that is it is positive. So, rate of formations of new cells Cc by dt. Okay. So, here after combining this equation or substituting this mu into equations, once we are substituting mu. So, then after substituting the monod equations for the exponential cell growth, we can write Rg equals to mu max Cs into Cc by Ks into Cs. This is the, if we want to want to know the in the exponential cell growth rate in a bioreactor for a particular specific cell reactions. So, here we can find out or we can use these equations to find out rate of new cell formations or cell means formations rate per unit times, that we can find out. Okay. So, if a conditions Ks is the that we said monod constant, that is the monod constant I said Ks is the monod constant. If Ks is very small, then Cs substrate concentrations, then this equation 3 we can simplify into this equations, Rg equals to mu max into Cc, cell concentrations. Okay. This is the cases. So, when Ks if we find out some value and Cs cell concentrations if you know, and under these conditions only this is applicable. Otherwise, this equation 3 we can apply or we can use to find out the cell growth, okay, in the bioreactor. So, here you can see the bioreactor cell growth with the substrate concentrations gradually increase. the growth rate, that is the expressed in terms of dc by cc by dt, that is cell growth rate, that depends on the nutrient concentrations. In the bioreactions you can see cell is there, cell concentration as well as substrate is there, two components are there. So, when the cell growth or Rg that we are talking about, that cell growth that depends on the not only cell concentrations, that depends only on the nutrient concentrations, okay, nutrient concentrations, and in that substrates. So, how many nutrients are there that we do not know. So, if there are more than one nutrients, that also depends on, means the growth, cell growth rate, that depends on the more than one nutrients concentrations also. So, then for many systems it has been observed that the products, I said already, that product that is formed during the reactions in the stationary phase. So, that product inhibit the growth of that particular means cell growth, that usually inhibits. So, therefore, these equations actually we can write, these equations we can write in this way. So, empirical form of monodification for the product inhibitions. I said in the cell reactions you can see we are taking cells and substrate, we are getting more cells as well as product. This product sometimes inhibit the cell reactions. So, in the cell reactions here cell reaction stopped. So, here means at this particular stage there is no new cell formations is there. So, therefore, at this particular stage we can say means cell formation is not there because of this maybe some other nutrient is, product formation is there, that product may be inhibit the cell growth. Okay. So, rate of growth. So, now empirical form of monodification for the product inhibitions, that we can include here, and that rate of growth equations we can write Rg equals to K observed mu max Cs by Cs into Cc by Ks plus Cs, this equation, where Ks is this, we can write here Ks equals to this, Ks observed equals to 1 minus Cp by Cp star. Cp star is the product concentration at which all metabolism ceases. So, we said that there is a cell reactions, product formation is there, that product concentration inside of the cell may be there, that has a particular product concentrations. So, at that particular product concentrations all metabolism means process inside of the cells, that would be stopped. Okay. So, that concentrations, if lower than that, then cell growth or cell formation should be there. Therefore, n is some empirical constant terms that is there. So, when cell production has stops or product production inhibits, in that case rate of formations of cell growth rate we can write this way. Okay. So, there are some other equations also for the cell growth rate, how we can express. So, this is the special cases, or these are the equations you can use depending upon particular cell culture or cell growth rate, how we can express. So, depending upon the particular reactions we have interest. So, these are the equation, also different type of equations for the growth rates that means available, so you can see also. Okay. So, here in this particular we once we talk about the cell growth in the at this particular phase there is cell death, means when the cells formation has stopped, after that cells will use that nutrient that is present inside of the reactors, but sometimes in absence of sufficient nutrients or maybe the product formation is there, that is inhibit the cell growth, then cell death is there. So, if we want to calculate or measure how much cell death means for rate of cell death means inside of the reactors in the stage 4. So, then rate of cell death that we can express in this way, Rd equals to Kd plus Kt into Ct first bracket into Cc. Cc is the concentration of the cell and Ct is the concentration of the substrate that toxic for the cell. Okay. So, I said the concentration of the substrate that present inside of the cells, that may be as a product or maybe some other substance that is present that inhibit the cell growth, or that is for death of causes for the death of the cells. And Kd and Kt, these are the constant also. Kd is called specific death rate constant and Kt is the death due to the toxic substance. Okay. So, these are the constant terms. So, we have to use here, and these are the two concentrations, means that is the substrate concentration that causes that is the toxics for the cell, and concentration of the cells that is present. So, then the rate law for the cell death that we can express, or equation 8 we can use to find out when there is a cell reaction is there in the bioreactor. So, rate of death of the cells per unit times in the stage force that we can calculate using these equations. Okay. So, in the cell I said that concentrations in the cell growth rate. So, here you can see that concentration of the cell and concentration of the substrate is very important. So, at the same times the temperature that we use for a cell growth, that temperature is also important. Means once we study for any cell culture for a bioreactor using a bioreactor. So, the reactions we can bioreactions we can conduct by changing at different temperature, let us say 30 degree centigrade, 40 degree centigrade, 50 degree centigrade. So, which one would be the optimum temperature for a particular cell reactions, that also we have to study, and based on that we can check that. So, which would be the optimum temperature where the cell growth would be the maximum. So, that way we can find out the optimum temperature for a particular system. Okay. So, thank you so much. So, today is up to this. So, we have discussed basically the monod equations for the rate law and different phases, that is means when cell we are culturing inside of the cell reactor. So, then how cell growth is theirs, exponential, and cell growth stops, and then cell death is there. So, these are the things we have discussed for the, these are the basic things we should know whenever we study more details about the bioreactors, bioreactor kinetics. So, then it is required. Okay. So, these are the things very important. So, here these are the two monod equations, monod equation for exponential cell growth, and this is the specific cell growth rate, that how we can express, and this is the combination of the two equations, that is the monod equation for the cell growth rate, that is very important, that would be frequently used, and also Rg equals to dCc by dt, that is the rate, that how we can express cell growth rate, how we can express, that is positive dCc by dt. Okay. So, these are the things we should know for the bioreactor study. Okay. These are the basic things. So, next day we will discuss something different for more bioreactor equations. Okay. Okay. Thank you.